Bismillahirrahmanirrahim. Welcome to my channel Science of Life with Dr. Muhammad Yusuf. Today we are going to start a new topic, that is the hematology. We are going to start. Today's lecture will be general functions of the blood. But before that, one ayah from Holy Quran. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم قل هذه سبيلي أدعو إلى الله على بصيرة أنا ومن اتبعني وسبحان الله وما أنا من المشركين कि आप कह दीजिए कि मेरी राह यही है। कौन सी राह है? कि मैं और मेरे मुत्तबईन, मेरी इत्तबा करने वाले अल्लाह की तरफ बुलाते हैं। अल्लाह की तरफ बुलाना मेरा और मेरे मुत्तबईन का काम है। कैसे बुलाना? अला बसीरतिन, यकीन और एतमाद के साथ। मैं भी अना व मन इत्तबानी, मैं भी और जिसने मेरी इत्तबा की, दोनों। व सुबहान अल्लाह, और अल्लाह पाक है। वमा अना मिनल मुशरिकीन, और मैं मुशरिकों में से नहीं। अल्लाह पाक समझ दे। दीन पे अमल करना और उसको दूसरों तक पहुंचाना, अल्लाह की बात को पहुंचाना, ये हमारा काम है। अल्लाह बात इसकी समझ दे। So we are going to talk about blood. Today's lecture will be only a general bird's eye view of the general functions of blood. Blood, as everyone knows, that is, blood flows everywhere in the body. There are some minute areas where there is no blood flow, but practically blood is flows everywhere throughout the human body, and it is responsible for supply of all the requirements of the cells. We have trillions of the cells, and every cell receives its nourishment. We have got all the nutrition outside our body. We take the nutrients and oxygen, all the requirements are around our body, in our external environment. But all the cells must receive, and it is the blood responsible for supply of all the requirements of all the cells of the body. And then blood is also responsible for the drainage of the metabolites. Metabolism is taking place in all these trillions of the cells, and every cell is producing waste products during its metabolism, and these waste products have to be got rid of. They have to be eliminated, and they are eliminated mostly through excretory organs, kidneys and GIT and lungs. So all these waste products have to be taken away from the site of production, the cells, to the site of elimination, that the different excretory organs. So blood is responsible for drainage and removal of all the waste products of each cell. And it is very important for the maintenance of all the internal requirements of the internal environment. It provides the internal environment. It is in fact the internal milieu interior. It is our internal environment. And this blood is being distributed to all cells of the body via with the help of the cardiovascular system. Heart is responsible for pumping of this blood throughout the body, all the vessels. Every cell has got in its vicinity blood vessels in which the blood is circulating, and all the functions are taking place across the capillary membrane. Now, blood itself, the physical characteristics of the blood, it is a fluid circulating in our blood vessels. The total volume of blood in human being, in an adult male, is about 5 to 6 liters of the blood, depending upon the size of the body, between 5 to 6 liters. And in female, it is slightly less, 4 to 5 liters of the blood is present in the female body. The temperature of the blood is about 100 degree Fahrenheit. 100 degree Fahrenheit or 100.4 degree Fahrenheit, about 38 degrees centigrade. Viscosity of blood, it is not as water. It is a thick fluid. It is 5 times more viscous than water. The pH, hydrogen concentration, pH of the blood is between 7.35 to 7.45. Average pH is 7.4, you can say. It is slightly alkaline blood. Color of the blood, it is red, but when it is oxygenated, the blood taking the oxygen from the lungs and circulating in the arteries, oxygenated blood is scarlet red. And when it is deoxygenated in the veins, and the blood is deeper red. Now, components of the blood, what are the parts of the blood? For this purpose, we take the blood from any vein, we put it into a test tube, and then we centrifuge, we withdraw the blood from the vein, and we centrifuge the blood, and by this centrifugation, the heavier parts will settle down and the watery part will be upper part of this tube. Is after centrifugation, heavier particles have settled down, and they are an upper portion is the fluid part, and this is the solid part. Formed elements, they are known as formed elements. So fluid part is the plasma, which constitutes about 55% of the whole blood, average. And this formed elements, most of which are the erythrocytes, red blood cells, they are 45%. Well, very small amount, 1% is that there is a buffy coat between plasma and this, which contains the leukocytes and the platelets. So total, this one is the 45%, and they are collectively known as formed elements. That is the platelets and leukocytes, and the most of this formed elements total volume is 45% of the total whole blood volume is the formed elements. So these are RBCs, WBCs, platelets, collectively known as formed elements. So these are the different components of the blood. We will see what are the other important constituents in the blood. Whole blood, as we said, it is composed of this plasma, 45%, we saw in the previous diagram. It is 45%, and it may range between the 46 to 63% plasma. And the formed elements, the remaining 45%, may range between 37 to 54% of the whole blood. The plasma is mostly water. 92% of the plasma is water, and then there are proteins, which are 7%, and then other solutes in the plasma, which are 1%. So this is the out of the plasma, the different components in the plasma. We will segregate these plasma protein and other constituents in next slide, inshallah. Formed elements, as we saw, there are 3 types of the formed elements. Red blood cells are erythrocytes, platelets are thrombocytes, and the white blood cells are leukocytes. Red blood cells are almost 99.9% of the formed elements volume is because of the red blood cells, and the platelets and white blood cells, they constitute less than 0.1% or even less than that. So these are the formed different components of the blood. Now plasma. Again, I'm going to the plasma, 92% water, and then the plasma protein and other constituents. We are going to segregate these into further classification. Plasma constitutes of 92% water, plasma proteins are 7%, and other solutes 1%. This is from the previous slide. Now, water is primarily responsible for transport. It is the fluid medium, which it is carrying all the substances in it, all the solutes, this protein, formed elements, all they are in swimming in this water. So water is responsible for transport of the many substances, organic or inorganic molecules, and formed elements, and heat. This also is carrying heat. Water is carrier of heat from the body. It carries the heat from the interior core of the body. It brings it on the surface to get rid of this extra heat. We will see it. We will discuss it, inshallah. So transport, so water is very important. It is soluble. Then the plasma proteins, which are 7%. There are three types of the important proteins. The most abundant protein is albumin, which is about 60% of the total blood. And so it is a protein. It contributes to the plasma osmotic pressure. And it is responsible for transport of lipids as well. It is responsible for transport of many hormones, steroid hormones and other hormones as well. So in combination with the protein, so albumin has got important function. We will discuss in one full lecture, we will take about the functions of the plasma protein. Then there is second class of protein, which are known as the globulins, 35%. Albumin, globulins, 35%, and they also transport the different hormones or different substances, and they are carried by the this globulin portion, but they are transport of the hormone. They also transport the lipids, but they are very important part of the immune system. We will discuss immunology as well, immune defense system of the body. These globulins are very important in that. And third group of the proteins is fibrinogen, which is only 4% of the total plasma proteins. That is, this is the essential component of the blood clot. This fibrinogen is converted into a meshwork of fibrin threads, which is for the sealing of any damaged blood vessel, for the prevention of the blood loss. So clotting should take place, blood clots, when it is exposed to a rough surface. So fibrinogen is responsible for this clotting. And then there is a small fragment of the plasma protein circulating in the plasma protein, which are maybe enzymes, maybe free hormones circulating in the blood. For example, this growth hormone or insulin circulating in the blood. So they constitute less than 1%. And there are different enzymes present in the plasma. So they are the regulatory proteins. So this is the plasma proteins, which 7% of the total blood. So rest is 1% left, only 1%. This 1% is not less important as it is only 1%. It is only 1%, but this contains very important group of the elements, electrolytes, are necessary for the vital cellular activity, which contribute to the osmotic pressure as of the body fluids, and major electrolytes like sodium and potassium and calcium and magnesium and chloride and bicarbonate and phosphates and sulfates. All these ions are in this 1% of the other solutes. So the electrolytes are present in this. Then there are other organic nutrients as well, not only the inorganic salts. There are organic nutrients used for ATP production, cell growth, and maintenance of the maintenance of the cellular functions as well. This includes lipids. There are fatty acids. There are the amino acids. There is glucose, that is carbohydrates, and amino acid. They are all this in amongst the 1% of this. Plasma proteins are the big proteins. They are the combination of different amino acids, and free amino acids are also present in the plasma, and fatty acids are also present. So these are the different components of plasma. Organic waste products are also present. They are carried, for example, carbon dioxide is present, gases are present, urea is present, creatinine is present. So carried to the site of excretory sites, and include urea and uric acid and creatinine and bilirubin and ammonium ions, etc. They are again the components in this other solutes. So this is very important component. 1%, this is very important components of the blood. So these are different constituents of the plasma. Now, function of blood, we are going to, as I said, this is only general functions of blood. Individual function we will discuss in detail, inshallah, next lectures, inshallah. Blood is responsible for transport. It is responsible for transport of every requirement of the cells of the body. It is responsible to the cell and every waste products from the cells to the excretory organ. So transportation of gases, carbon dioxide and oxygen and Nutrients have to be carried to all the cells of the body. Hormones produced in different parts of the body, and hormone is being produced by the pituitary, and my muscles need the hormone, so blood is responsible for the carriage of this hormone. Metabolite weights have to be transported from all the cells of the body to the excretory organs, and then this is the transport function. These are the important substances which are transported. There are other. Iron is transported, carbon, glucose is transported, every transport of the different nutrients, different electrolytes, and many other substances. Then regulation of pH. This another very important function of the blood is regulation of pH. It has got an important buffer system in the body we are seeing. Hemoglobin itself is the biggest buffer in our blood. That is, hemoglobin proteins are buffer. There are other buffer systems in the body which are responsible for the maintenance of the normal pH. You must be knowing that the maintenance of normal pH must for the proper metabolic. Increase or decrease pH disrupt the enzyme system, then the metabolism depends upon this enzyme system of the body. So main regulation of pH is another important function of blood. Then regulation of electrolytes and composition of the interstitial fluid. That is the main components of the extracellular fluid and intracellular fluid, interstitial fluid, plasma. The different constituents have to be regulated. They have to be precisely within the normal limits, and this is the blood itself which is responsible for this regulatory functions. Then blood is responsible for the prevention of extra fluid loss. If some bleeding occurs, prevention of bleeding is again the function of the blood. Clotting mechanism, for example, we are going to discuss. Or the restriction of the fluid loss, diarrhea and vomiting, and blood plays its important role for the maintenance of the different fluid compartments of the body. So restriction of the fluid loss from the body, blood plays its role. And then immunology, defense system of the body, defense against different pathogens, different bacteria, different viruses, or different foreign invaders. So defense against the toxins and pathogens. This is again the part of the blood and the function of the blood. Then blood is also very important for the regulation of the body temperature as well. As I said, heat is produced in metabolism, and this heat has to be got rid of from the body. And when extra heat has to be got rid of, and when the body is exposed to the cold atmosphere, then conservation of heat is required. So blood also plays its role for the stabilization of the body temperature. So this is the list, important functions, the list of the important functions of the blood. Now, blood and transportation of, as I said, oxygen is mostly carried by hemoglobin. Hemoglobin in RBCs, in red blood cells, the most abundant formed elements in the blood. RBCs are erythrocytes which contain hemoglobin. Oxygen from the lungs to all tissues of the body is carried by hemoglobin. This is the shape of the RBC. They are biconcave discs. They are responsible. They contain hemoglobin in them, and hemoglobin combines with the oxygen, and this is carried by the hemoglobin in the blood. So carbon dioxide produce different metabolism is carried to the excretory organ, that is the lungs, alveoli of the lungs, to get rid of in the expired air. So carbon dioxide is carried in combination with the hemoglobin as well. And dissolved in the plasma, carbon dioxide, but mostly in the form of bicarbonate ion. Carbon dioxide is converted into combines with the water and form the carbonic acid, which dissociates into bicarbonate ion and the hydrogen ion. So bicarbonate ion. So these are the three forms are dissolved in plasma protein and combination with the hemoglobin. Some carbon dioxide may be carried in combination with the other proteins as well. So gases are carried by, and nutrients Nutrients are carried from GIT. We take the nutrients in the form of our diet. After digestion and absorption of all the nutrients, carbohydrates and proteins and fats and all, they are carried by the blood from the GIT to every cell of the body. And then there are some nutrition stores present in our liver. Liver is the rich storehouse for the different nutrients. Proteins are synthesized there, they may be stored there. And so from the liver, different nutrients, glycogen is stored in the liver. So that glycogen may be taken by the blood to the, if they are, this glycogen is required by some other muscles, so liver. And the adipose cells are fat cells. Adipocytes, fat cells, they contain the stores of the fats. So fat, when it is required, fat is to be utilized. So fat can be from this storehouse. And from the muscles, one another thing, muscle are also the storehouse of the protein. If proteins have to be supplied to the other parts of the body, muscle are the storehouse for the nutrients. So all the nutrients, either fresh from the GIT or from the storehouses of the body, that is the liver and the adipose cells and the muscle cells, blood takes these nutrients from this and supplies all cells of the body. Then hormone produced by the different endocrine glands located in different parts of the body, and these hormones may be required by different cells of the body, all cells of the body or some cells of the body. So blood will carry the hormone from the site of production, endocrine cells, to all the cells which they require. Then the metabolic waste products are transported, as I said, carbon dioxide or creatinine or uric acid or urea produced in the metabolism. Metabolites are carried by the blood. So these are the transport functions of the blood. Regulatory functions. Blood is regulating what? Regulation of the temperature, for example. A very crude example I have taken. This is the cold atmosphere. Blood distribution is, blood remains in the core on the surface. Vasoconstrictions occur. Less blood supply on the surface causes less wastage of heat. So heat is conserved. But in the warm weather, when extra heat is present in the environment as well, and metabolism is also producing, or the person is performing exercise and there is lot of metabolism and lot of metabolites produced and lot of heat produced, so how warmer body, blood is distributed throughout the body like that diagram, and this can be got rid of into the environment. So for the regulation of temperature, blood distribution becomes important. So relative distribution of blood in response to warm and cold temperature is visible in this diagram. Then pH regulation, as I said, blood has got the important buffer system of the body. Hemoglobin is the most important, biggest buffer in the blood, which maintains pH. It prevents the extra rise or fall in the hydrogen ion concentration. Extra rise is the increased hydrogen ion concentration is the acidity, and decreased hydrogen ion concentration is the alkalinity. So this hemoglobin provides the buffer. We are going to discuss how it provides the buffer action, how it is the buffering power कैसे achieve होती है. So we will see. And there are the plasma proteins. They are also important in the buffer mechanism. There are other buffer, bicarbonate buffer, then the phosphate buffer, then we will see. Different buffer systems are present in the blood, which are important for the maintenance of pH. Then electrolytes have to be controlled, sodium level or chloride level, and distribution of this in across the this capillary membrane. Inside the cell, potassium is more, outside the cell, sodium is more. So blood is responsible for the distribution and the maintenance regulation of this. And the blood volume itself, which is very important for the blood volume circulation. If you have seen my lectures on circulation, they are available on my site. That is the blood volume, how the blood volume is regulated and blood pressure is regulated. So blood pressure regulation, blood volume regulation. Then the defense system of the body, very important. Defense against the foreign invaders. Immunity, which we term as immunity, we will discuss it, immunity in detail. It is a long discussion. Different types of immunity are present in our blood, provided by our blood. So immunity, and then the prevention of blood loss Clotting mechanism is another very important mechanism, clotting of the blood. For example, whenever there is damage to the vessel, then the blood clots to seal that damage. Clotting must take place. So it is clotting is the function of different interaction of the plasma protein, and finally the fibrinogen is converted into fibrin network or meshwork, and the RBCs are entangled and blood clots. It is converted into semi-solid state. So it seals the damage in the blood vessel. So this clotting is also important. Images of the blood clot. This is image of one blood clot. Formed elements we will discuss. Although formed elements separately, WBCs, RBCs, etc. Formed element, there are three types of the formed elements, are cells. One is the leukocytes. They are responsible for the defense system, other functions as well. Leukocytes, and they protect against diseases, and they are police force of the body. They are against the pathogens, against the invaders, and they provide immunity, the defense system. There are types of the white blood cells, different types of the blood cells. There are two major types, but 5 or 6 types of the blood cells, white blood cells are there. There are some cells, white blood cells, which contain granules in their cytoplasm. They are known as granulocytes. There are granules in the cytoplasm. They are known as granulocytes. And there are cells, white cells, which are, they don't contain granules in the cytoplasm. They are known as agranulocytes. A stands for no granules. So agranulocyte. Granulocytes are 3 types, depending upon the staining reaction of those granules. There are the most abundant cells, which the granules of which take the neutral stain. Usually we use the stain, HNE stain is the most commonly used stain, HNE, hematoxylin and eosine. Hematoxylin is the basic stain, and hematoxylin is the eosine is the acidic stain. These cells, neutrophils, which are neutrophil, the granule take the neutral stain, both mixed acidic and basic stain, neutral stain. So they are known as neutrophils. Then there are cells which contain that acidic stain, eosine. They are known as eosinophil. Philia means love. They love eosine. They attract eosinophil. And then there are cells which take the basic stain. They are basophil. They are stained by hematoxylin, the basic stain. They are known as basophil. So these are 3 types of the granulocyte, but they are WBC, leukocytes. And there are agranulocytes, lymphocytes, which are very important part of this immune system of the body. They are also involved, but the very important part is the lymphocytes, which provide the antibodies. The main defense system of the body is by the lymphocytes. So lymphocytes are amongst the agranulocytes. Their cytoplasm doesn't contain any granule. And then there is fifth type, the monocytes. They are also the WBCs. We are going to see the diagram of all these cells in next few slides. So there are 2 types of the two classification of the WBCs: granulocytes, agranulocyte, depending upon the staining reaction, divided into neutrophils or eosinophils and the basophils. And depending upon the agranulocytes are 2 types. They are lymphocytes and monocytes. Types of leukocytes we are going to see. These are 3 granulocytes. They are collectively known as polymorphonuclear granulocyte. Granulocyte cytoplasm contains granules. This diagram is not good. Next slide is good diagram is also available. But these at the same time, there are granules in the cytoplasm. This is the nucleus. This blue part. This is nucleus. This is nucleus. Nucleus is mostly composed of chromatin, DNA. DNA is acid. It will take the basic dye. it will be stained by the basic dye because acid will combine with the basic, that is hematoxylin. Nucleus is usually blue shaped. Blue. So, but this is all nucleus, but there are many shapes of the nucleus. They are known as polymorphonuclear. Poly means many, morpho means shape. Nucleus has got many shapes. So nucleus has many shapes in all these three cells, but depending upon their staining reaction, these are neutrophils. Granules will take the neutral dye. Granules will take the red dye, eosinophil, and they are known as eosinophil. And granules and nucleus both will take the basic dye. They are basophils. So these are the granulocytes. And the agranulocytes, they are mononuclear, not like this, the polymorphonuclear. Mononuclear, this is monocyte, the biggest one, and that is lymphocytes. Detailed, good diagrams are in next slide. Granulocyte, this is a good diagram of neutrophil. This is neutrophil. Neutrophil, these are RBCs. This is the slide. These are rest of that. These are RBCs, biconcave disc. We are going to discuss. This is a neutrophil. Many shapes of the nucleus. Nucleus may be 1 lobed, 2 lobed, 3 lobed, 4, 5, 6 lobes. As the age advances, neutrophil gets older, the number of lobes of the nucleus increases. So this is 1, 2, 3, 4, 5 lobes are probably visible, 5 lobe nucleus. But the granules in the cytoplasm, they are taking the mixed red and blue, violet color. They are fine granules and violet color granules, so this is neutrophil. This, another cell, nucleus is bilobed. And the granules are taking the red stain. This is the violet color stain, this is the red stain, so this is known as eosinophil. This is eosinophil. And if the granules take the blue dye, and this is basophil. Blue dye, no, nucleus also takes the blue dye because of the DNA, and these granules also take the blue dye. So boundaries between the cytoplasm and nucleus are not clear as it is here. It is clear boundary between the nucleus and the cytoplasm. Here it is mixed because the nucleus is also taking the basic dye and the granules are also taking the basic dye. This is the basophil. So these are the granulocytes. So out of those 5 cells, these 3 granulocytes, this is basophil. And the agranulocytes are the lymphocytes and monocytes. So this is a monocyte. The granules are, this is not, this is some staining reaction, something wrong. This is monocyte, kidney shape. Usually the nucleus is the kidney shape and there are no granules in the cytoplasm. These are the RBCs. And so this is monocyte. And the lymphocytes are usually whole of the interior is filled by the nucleus. Only small rim of cytoplasm is left, which is agranular, no granules present. So it is the lymphocytes. It is slightly bigger than or almost the size of the RBCs. Lymphocytes are there. So these are the 5 types of WBCs. This is lymphocytes. Now, plasma protein, as we saw in the previous slide, we saw that there are 3 types. The total concentration of plasma protein is 7%. We saw that between 6 to 8 grams per deciliter by weight. If you measure it in the grams, it comes to 6 to 8 grams per deciliter total concentration of the plasma protein. And there are types we have seen: albumin, globulin, and fibrinogen. These are the 3 types of the protein, different functions we are going to discuss. We can simply list the functions of the plasma protein in detail in next lecture. We are going to take the next lecture, inshallah, on the functions of the plasma protein, just listing of the different important functions. They provide colloid osmotic pressure of the plasma. This is very important pressure in the circulation. Colloid osmotic pressure, and they are important buffer for the pH, maintenance of the pH. They transport the hormones. Plasma protein transport the metals, iron and copper, cobalt. that is taken by in combination with the plasma protein. And blood clotting factors, mostly there are many clotting factors. Final clotting is due to the fibrinogen, but there are many other plasma proteins which act as the clotting factors. So blood clotting is the function of the plasma protein. And then prevention of clotting. Blood should not clot in our circulation. So prevention of clotting is also very important anticoagulant functions of the plasma protein. Then they provide nutrition. Nutrition is provided to all cells of the body. These proteins may be utilized by all the cells of the body, so they are important nutrients. And then next, they are part of the very important functions of the plasma protein, immunoglobulins, that is, they form the antibodies, they are part of the immunity, they provide immunity. And they contribute toward the viscosity of the blood. The blood is 5 times viscous than the water, so they contribute toward the viscosity of the blood. Then there are enzymes circulating in the blood, and they have got the respective functions. Enzymes, we will see, but important enzyme, and due course we will discuss. There are enzymes in the plasma protein. And then the hormones circulating in the blood. Growth hormone is a protein which is circulating in the form of the plasma protein. Insulin is a growth hormone, and there are many anti-pituitary hormone, parathyroid hormone is a protein. So they are part of the plasma protein hormone circulating. And then the angiotensinogen, very important protein present in the plasma, which is converted into angiotensin, and that is very important for the regulation of the blood pressure and for the regulation of the electrolytes as well. So renin-angiotensin system, angiotensinogen is one of the plasma proteins. So different functions of the plasma protein we are going to discuss in next lecture. A few things about the red blood cells, basic concept about the red blood cells. Erythrocytes, which are known as the erythrocytes, they are like biconcave discs. They are discs. They are thin in the center and thick at the center, like biconcave discs. You can see the diagram. And the most abundant cells in the body. In our whole human body, most abundant cells are the red blood cells, and their count in our blood is about 5 million per cubic millimeter of blood. 1 microliter of blood. 1 cubic millimeter of blood is 1 microliter. 1 microliter contains 5 millions of the RBCs, plus minus 300 maybe. This is the physiological range. You can add or you can subtract. Normal is, average is 5 million. In male, in female they are slightly less, 4.7 million per cubic millimeter of blood. And they contain red pigment, as in previous slide we saw. That is, they contain hemoglobin, red pigment hemoglobin, which is responsible for the carriage of oxygen as well as carbon dioxide. Each RBC is a biconcave disc, and diameter of our RBCs, this diameter is about 7.5 to 8 micrometer. And the thickness in the periphery is 2 to 2.5 at the periphery, and in the center only 1 micrometer. 2 micrometer in the periphery and 1 micrometer in the center biconcave. Center में thin होते हैं, periphery पे thick होते हैं. This is the how. This is the RBCs. Total diameter is 7.5 micrometer. And this is the side view, that is in the periphery. And this is the 2 micrometer in the center. It is only 1 micrometer. So erythrocytes, why they are biconcave discs? When the cell is biconcave, it is concavity on those sides. Total surface area of the cell membrane is increased. So this provides a large surface area for the exchange of gases. Large surface area. Oxygen has to diffuse through this cell membrane into the cells to combine with the hemoglobin. So it increases the surface area, number one. Then it enables them to bend and flex. They are loose bags of cytoplasm containing hemoglobin. They are loose bags. So this biconcavity and there being absence of nucleus, there is no nucleus in the RBCs. This provides the easy flexibility, that it enables them to bend and flex in small blood vessels. like this. This is RBCs. This has bent into, this is a capillary, small capillary. This is the nucleus of the capillary cell, the endothelial cell. This RBC is passing through a small capillary and it has bent itself into it. It can squeeze itself. So this provides this biconcavity, and the looseness of this cell membrane provides this easy flexibility of the cell. So RBC, they bend to fit through the small capillaries. And the erythrocytes, RBCs lack nucleus. There is no nucleus. And many other organelles are also absent. So that's why they have got the limited lifespan. They have got the lifespan of only 120 days. So they lack nucleus, and they are the membranous bags of cytoplasm containing a large amount of hemoglobin. 35% of the interior of the cell is hemoglobin. So simply a membranous bag of hemoglobin and water. And it can deform, it can change to any shape, it can deform. And because of this, deformability is very important quality of the RBCs. So they can squeeze through small pores. With this process of squeezing through the small pores, as we saw in the previous diagram, they can squeeze through the small capillaries. Again, another diagram coming in next slide. So this process is known as diapedesis. They squeeze themselves through small pores, diapedesis. This is a slide of the alveolar epithelium. These are the alveoli, and this is the capillaries, and RBCs are passing through the capillaries, and they are adopting many shapes when they are passing through the capillaries. All these are the RBCs passing through the small capillaries and deforming. This is diabetes. They can squeeze themselves through the small capillaries. Now, the lifespan of RBCs, as I said, is only 120 days, and there is no nucleus, no DNA, and no RNA for the normal control of the nucleus. Is responsible for the survival of the cells and controlling the function of the cell, providing all the requirements, and so the nucleus is absent. So there is 120 days lifespan. And they cannot synthesize the protein for the repair and growth because of the absence of this. And the irreparable cell membrane becomes fragile with the passage of time, and then ruptures when it passes through the small spots, especially in spleen. स्प्लीन में स्मॉल स्पॉट होती हैं. जब membrane becomes fragile because of the no repair taking place because of the absence of nucleus, they are removed from the blood by the spleen. So this RBC, they provide viscosity of the blood. Why the blood is 5 times more viscous? Because of mostly due to the RBCs. RBCs come more, the blood becomes less viscous. Anemic person have got the less viscous blood. So the increased number of RBCs increases the viscosity of the blood, and decreased number, as we will see in anemia, it decreases the viscosity of the blood. Hemoglobin, this is hemoglobin carries, it is the carrier of oxygen and the carbon dioxide. And carbon monoxide may be carried, which is the poisonous gas, carbon monoxide, but it can combine with the hemoglobin as well. Nitrous oxide, when it is required, it is given in anesthesia, NO, that is given. So this is nitrous oxide can be carried by the hemoglobin. Hemoglobin is the biggest buffer in the blood, and the carbonic anhydrase is enzyme present in the RBCs, which is important for the reaction between the carbon dioxide and water to form the carbonic acid, and then it splits into hydrogen ion and bicarbonate ion. So the carbonic anhydrase is an enzyme present in the RBCs. This reaction, that is carbon dioxide plus water, leads to formation of carbonic acid, which can dissociate into hydrogen ion and bicarbonate ion. This is the function of carbonic anhydrase. Hemoglobin is a big protein we will discuss in detail. It is composed of 4 chains, 2 beta chains and the 2 alpha chains. This is the structure of hemoglobin. It contains heme. Iron containing portion is the heme. We will discuss in detail that is hemoglobin. RBCs contain hemoglobin. Some special values about the RBCs should be on our fingertips, which we term as the absolute values for the RBCs. Total count, we have seen it is 5 millions per cubic millimeter of blood. In adults, male, and in female, it is 4.7 million per cubic millimeter of blood. Size of the RBCs, we saw that the diameter is about 7 to 8 micrometer, outer edge thickness is 2 micrometer, and the center thickness is 1 micrometer. And hemoglobin contents of each this hemoglobin, 34% of the cell volume. Cell volume ka 34% is hemoglobin. But in that is, if you measure the hemoglobin contents in whole blood, not the individual cellular mass, whole blood, it out of 100 ml of blood, deciliter of blood contain 14 to 16 grams, 14 to 16.5 grams. In 100 ml of blood, whole blood, is hemoglobin, 14 to 16 grams. Usually, this hemoglobinometer, which gives you the, you measure the hemoglobin in this form, 14 gram per deciliter, 15 gram per deciliter, and if it is less, it is 10 gram or 9 gram per deciliter. This is per deciliter, 100 ml of blood. In female, it is less, 12 to 15, and in male, it is 14 to 16 gram per deciliter, normal values. So RBC hematocrit is the percentage of farm element. The first slide we saw, when we centrifuge the blood and the total percentage of the farm element in whole blood, that is known as hematocrit. That is, it is in male, it is 40 to 50%, 45% we saw in that diagram. In male, it is 40 to 50%, and in female, it is 37 to 47%, slightly anemic in female. And oxygen carrying capacity total, how much oxygen can be carried by this hemoglobin or RBCs? That is 1 gram of hemoglobin. Normal person contains 14 grams, 16 grams, previous slide. 1 gram of per deciliter. 1 gram of hemoglobin can carry 1.34 ml of oxygen. 1.34 ml of oxygen. If the 100 ml, which contain 14 grams or 15 grams, 100 ml of blood, you multiply this with 100, it will be carrying about, and if a male, it is about 20 ml of oxygen is carried by the male 100 ml of blood, and in female, 19 ml of oxygen can be carried by the 100 ml of blood in female. MCV, it is mean corpuscular volume, volume of one cell. एक सेल का वॉल्यूम कितना है? It is 100 to 100 femtoliter, 80 to 100 femtoliter, and MCH, mean corpuscular hemoglobin. How much is hemoglobin in one cell? It is 27 to 34 picograms. In picograms, we measure MCH. And then another value is important, MCHC, mean corpuscular hemoglobin concentration, percentage of hemoglobin in this cell, mean cell. It is about 31 to 35%, पहले भी कहा मैंने 34% होता है, between 31 to 35 grams per deciliter. If you take 100 ml of this deciliter is 100 ml. 100 ml of RBCs, only RBCs, out of that will be containing 35 grams of hemoglobin. So that's all about the this bird's eye view of the functions of the blood. We will discuss all these functions in detail, inshallah. Next lecture, inshallah, our lecture will first we will talk about the functions of plasma protein in next lecture. Hope to see you next lecture. Please do share and subscribe to my channel and share this to the rest of the students so that most of the students can benefit from it. And it will be sadqa jariyah for me and for you as well if you share to other students as well. Thank you. Hope to see you in next video, inshallah.